Beside for you taking a stethoscope and hearing the bowel sound and know that we have a rate of about 15 to 30 per minute, unlike the heart rate. And other physical examination, like you can do a percussion, you can do some deep palpation and shallow one. Besides that one, you don't have so much to talk about, but we have a lot of physiological process. Also, over your course of learning, you have been interacting with the digestive system for long, and we are only going to add a few things that are of significant in terms of physiology. On the same note, just bring back your anatomy in a review. And this is simply a tube, a long tube that runs all the way from your mouth and end all the way at the anal. And that tube is made up of several parts, as you already aware of that one. So then, what are the function of the GIT system? So it's a source of nutrient, it's a source of electrolytes, and it's also a source of water. Anything we take through the mouth, it will have to go through the GIT, and later the body absorb it. That's the primary function, and which of course is essential for the maintenance of life. So the input of food involves some movement throughout the GIT, and through that movement will end up with digestion, and the nutrient will be distributed throughout the body. And just like any other system, it doesn't work in isolation. It has to be under the control of the nervous system, together with the control of the endocrine system. Those are the hormones. You still remember that I said we have some few hormones that we didn't cover in endocrine system, and we reserved a few of them. We'll still be mentioning some of them here. Because it's made up of smooth muscles, I said that anything that is circular or cylindrical in the body is always made up of smooth muscles. And these smooth muscles are arranged in 2 layers, or sometime in 3 layers in a few areas, as we'll see. We have the longitudinal and the circular one. And we have the internal mucosa, which is an epithelium, either adapted for digestion or for absorption. And we have the serosa, together with the connective tissue that supports the structures. The resting membrane potential, that's another term that I'll introduce when you talk about the muscles later. But just know that the GIT system is capable of relaxing and contracting on its own without the influence of the brain. We call that one an intrinsic ability to contract. But at the same time, the brain can still influence it through some branches of the autonomic nervous system called either the sympathetic or parasympathetic. And we'll later see that the parasympathetic is the major dominant branch in the GIT. That's what enables you to sit right now comfortably and digest that food. As a product of movement, move all along from your stomach into the large intestine. We have a branch of it called the enteric nervous system, and it's a branch of the autonomic nervous system that functions independently. It's made up of 2 plexus. We have what we call the myenteric and the submucosa. These 2 terms are simply used to describe their location. So their function, the myenteric one, which is located between the muscles layer, once we have a stimulation of it, it's almost the same as stimulating the parasympathetic nervous system. The aim is to increase the process of digestion, and through the following ways of increasing the tone, increasing the rhythm and intensity of contraction, and at the same time, the velocity will increase and relaxes the sphincters. So the submucosal plexus, also called the Meissner corpuscles. So that one is located between the muscles and the submucosa. And the function is a sensory input from epithelium, and later it controls the secretion and absorption of the GIT. So the autonomic nervous system, as I said, we have the sympathetic and parasympathetic. I'll reserve that one until when we cover about the nervous system. We have a region where it originates and where it innervates. So for now, kindly let pass through that one. We have reflexes in the GIT system. So this functions within the enteric nervous system. and it functions to control secretion. This is the secretion of all the enzymes requires and some other substances, the movement within the JT that we call it peristalsis, and also the inhibition of the same movement. So we have what we call the reflexes of the gut, so which happen through the sympathetic reflex. So we have the gastrocolic. So the first word indicate where the reflex start, and the last word indicate where it ends. So from the stomach, the gastrocolic reflex start from the stomach, and it goes all the way to the colic, that is the colon. And the function is to ensure that we have a contraction of the stomach once we have the emptying of the colon. And then we have the enterogastric. So it started from the intestine and goes into the gastric. So the function of that one is to inhibit the emptying once the colon is full. So from the gut into the brainstem, these are the functions of the nervous system of either branches. So we call that one either sympathetic or parasympathetic. And it enhances all these processes, such as the process of defecation, that is simply emptying the colon, or the control of all other movement associated with the JT. These are the hormones available that I've been mentioning about them. We have the secretin, gastrin, cholecytokinin, gastric inhibitory peptide, and motilin. So the function is either to stimulate or inhibit the JT movement. We have two types of movement. We have the propulsive movement. So the movement of propulsion is to ensure that the content, in this case the food, either digested or semi-digested or fully digested. And the process of mixing, what you call the chiming sometime. So this movement occur because of the reflexes that I said. We call it the myenteric reflexes within the GIT. We have a principle called the law of the gut, and that one is concerned with the process of peristaltic movement. So the direction always move from your mouth towards the anus, and that one ensure that we have one directional movement. And then we have the mixing that enhance in the process of digestion and absorption. So we have the splenic circulation. This is simply the blood supply to the GIT, the pancreas, liver, and the hepatic. So we have the hepatic vein, which supplies blood, and usually that one is after the process of digestion, as we'll see later. The propulsion and the mixing of food is the other type of movement that we said occurs through the following phases. So once we have the ingestion of the food, we have the pharyngeal stage, which is usually immediately you take your food and put it into your mouth. So you chew it and then you start swallowing. Swallowing occurs in the following process. We have the voluntary part of it, and then we have the involuntary part of it. So the voluntary part is the one that you have conscious control of it. Your attempt to swallow is an involuntary process, and from there it becomes automatic, and that one becomes involuntary. So once the food is at the esophagus, it becomes automatically involuntary under the control of the nervous system. We'll see the nerve specifically involved in that region later in our nervous system section. But we have the primary, secondary, peristalsis. So that movement will go, the bolus of food will go until it encounters a sphincter, and the sphincter will relaxes and the food will enter the stomach. Once we are at the stomach, we have the following function. We have the motor aspect part of it. So that entails storing the food temporarily, and later it has to empty. And we have that mechanical movement or mixing, we call it chime. That control is under the brain, what you call the vagovagal reflex. So the vago only means a nerve that you call it the vagus nerve. We'll talk about it in the nervous system in detail, but it's cranial nerve number 10. So we have the sphincters there. We have one on the upper side called the cardiac, and we have the one on the lower called the pyloric sphincter. So both of them are under the influence of hormones to either close or open depending on when the food is needed to move into the small intestine. And we know the first part of the small intestine is the duodenum. So at the duodenum, we have what we call the segmentation contraction, that a contraction of any sequence at a velocity of about 5.5 to 2 centimeters per second. So the control of peristalsis is still under the control of the nervous system and hormone, just like any other part of the GIT. We have the sphincters, so called the ileocecal sphincter. So this is between the ileum and the cecum, near the appendix. And that's the region usually where we have the highest bowel sound. And once you are checking the bowel sounds, for example, a patient has been done an operation, and before the patient, we advise them to start taking meals. We have to ensure that, or at least have evidence that the patient is having some GI activity. We take a stethoscope and auscultate on the left side of the lower quadrant. That is the approximate region where we have that ileocecal sphincter, and that's where we have the maximum bowel sound. Another way, the simplest way, is simply to see whether the patient is able to pass flutters, because ability to pass flutters means we have the activity of the GI. All right. After the small intestine, we go to the colon. The functions include the absorption of water, electrolytes, and the storage of the fecal matter. So we have, instead of the segmentation contraction that we had in the small intestine, we have what we call the haustral contraction. And the purpose of that one is to enhance propulsion and the mixing. We also have the mass movement. Mass movement is involved in the clearance of the content of the colon. And then we have these gastrocolic reflexes and duodecolic reflexes, which communicate back to the stomach and to the duodenum respectively, depending on the content on what is available in the colon. An example in babies, for example, of the gastrocolic reflex is once a baby, they feed immediately, they have a desire to pass out stool. Is because of these gastrocolic reflexes, because the stomach is full, and that one sent a reflex into the colon to cause a contractions in the colon, and the fecal matter or defecation process happens. So yes, this is the defecation process. It's an intrinsic process, and it involves the internal anal sphincter and together with the external anal sphincter. And I hope you just remember from your anatomy that internal anal sphincter is smooth muscles, and anything that's made up of smooth muscles is under involuntary control. And the external anal sphincter, which is about the 2/3 of the anus, is made up of skeletal muscles and being supplied by the pudendal nerve. And you can have a conscious control of that one. And for that reason that, you want to go into the washroom, you can feel pressure a little bit, but you can still be able to hold it up. Correlate that one with what we did in the urinary system, where we said we have the internal sphincter, which is made up of smooth muscles, and the external one, which is made up of skeletal muscles. And that one, the skeletal muscles, you have a conscious control, you are able to regulate it. So, and we said that parasympathetic is all control the defecation reflex. Remember the term that we said that parasympathetic is synonymous with digestion and sleeping. And all these reflexes happen at the brain level, but we can modulate them from the higher brain centers. So that was about the movement of the GIT. Let's go into the second phase and see the secretory functions of the GIT. So secretion, we talk about the glands. We have several of them that start all the way from your mouth, where we have the three salivary gland, we have the parotid, sublingual, and submandibular that secrete saliva. It secretes the digestive enzymes and the mucus for lubrication. Those are the two purposes. And it's under the regulation of both the hormonal and the nervous system. So the stimulation can happen using a tactile. So for example, when you put something in your mouth and you start rolling it like a flat, something rounded, or simply chewing your pen, for those people who have a tendency to chew their pens. So that one induces the secretion of the saliva. So it could also be because of chemical, or it could be just because of the distension of the wall of the stomach. It enhances the secretion of the enzymes. Parasympathetics ensure we have sufficient secretion, but once you activate the parasympathetic, it does the opposite and cancel the function of the parasympathetic. Let's talk about the saliva. We said it's being secreted by the three salivary glands, and the active part of it is called ptyalin or alpha amylase, and the function is to start digesting starch. And the process of digestion starts immediately in the mouth all the way until the food reaches the stomach. Once you are at the stomach, what happens? At the stomach, we have the glands, and these glands secrete the following. So we have some cells types in those tubular glands, around 3 of them. We have what we call the chief cells that secrete pepsinogen. So pepsinogen is an inactive form of pepsin, and the activation is done by the hydrochloric acid and the HCl being secreted by the parietal cells. And it's distributed along this region of the stomach. The function of secreting pepsinogen in an active form is to ensure that it doesn't digest the cells that actually secretes it. That was the function of the HCl, is to ensure that it activates pepsinogen, and also to ensure that it gives a very acidic environment to really neutralize any other pathogen that might be available in the food. The secretion is under the control of the following phases. We have 3 phases of them. We have the cephalic, the gastric, and the intestine. So cephalic means brain, gastric means stomach, intestinal simply means the intestine, in this case, the small intestine. So just next to it, we have the pancreas. We know it has 2 roles. We have the exocrine part of it and the endocrine part of it. We did handle the endocrine part of it extensively when we talk about the alpha and the beta cells that secrete glucagon and insulin for the regulation of sugars in the body. In this case, we are interested in the exocrine part that secrete the pancreatic juice, which helps in the digestion of protein. It's made up of the following trypsin, chemotrypsins, and all the way, and the name indicate on what it digests. So the main function is to digest the carbohydrate, and that's a function of the pancreatic amylase. So it also hydrolyzes starch, convert it from glycogen into the glucose form of it, and all these other enzymes like the pancreatic lipase, the cholesterol esterase, and a phospholipase. So the function from the word indicate what function it does. So for example, the cholesterol esterase hydrolyzes cholesterol esters. We have the epithelial cells that lines the ducts, and these epithelial cells secrete bicarbonate. So the function of that one is to neutralize the HCl. We know near the pancreas, just adjacent to it, we have the liver, the biggest solid organ in the body. And in this case, we are interested in the digestive aspect part of it. Within it, we have the gallbladder, which directly open into duodenum. And the function of the bile is to enhance the process of emulsification that later will lead into the digestion of fat. And you know it's stored in the gallbladder, and the secretion is under this hormone called cholecystokinin. And the function of storing temporarily at the gallbladder is to ensure we have concentrated it into highest concentrate possible, such that a very small volume will be required for digestion. At the small intestines, that is the ileum, all the way from duodenum until the end part of the ileum, we have the Brunner glands. These are simply the mucus secretory gland. And we also have the craft of Leberkan, and these are goblet cells also that secrete mucus together with a few electrolytes absorption. Finally, at the large intestines, so this is the colon, both all the way from the ascending, transverse, and descending, and the sigmoid colon. So we have an extensive secretion of mucus. The purpose is to prevent an abrasion, or what we call the exocretion by the fecal matter into its internal lining. And it's also form a protective layer against the bacteria. So the bacteria is mostly the normal flora, and the purpose of this bacteria is to break down the undigested part of the food that you ate. So for example, the starch and that is hard to break it will be broken down by this bacteria in the colon. So the third part is for us now to see the actual process of digestion and the macromolecule involved. So we'll talk about the carbohydrate, fats, and protein, and what electrolytes and vitamin don't require absorption, so don't require digestion. They will be absorbed directly. Let's look at each and every of them. Carbohydrates start all the way from the mouth, and this is where salivary amylase will act upon the starch, and it will continue until it reaches the stomach. And because of the change in acidity or in pH from alkaline into acidity, it will inhibit the activity of the salivary amylase, and the digestion stops. So we know glucose will form the final end product of all carbohydrate. And we have the three macromolecules of carbohydrate. We have the lactose, which is a combination of galactose and glucose. And once you break it down, you end up with the two molecules. Lactose, we know we get it from milk. We have the sucrose. This is like the table sugar that you use, and it's made up of glucose and fructose. And finally, we have the maltose, also sometimes available mostly in cereals. It's made up of two molecules of glucose. And once you digest, it's simply we are reversing the process that made up them. Proteins, so that digestion starts in the stomach, where pepsin will act on it and break down the collagen fibers. And from there, other enzymes from the pancreatic, from the pancreas will act upon it. We have the proteolysis, we have the peptones, polypeptidase. So the aim is to break down the protein into the final building products, which are amino acid. And this is what can be absorbed from the small intestine. And we have what we call the enterocytes that lines the small intestine. Even inside the enterocytes, digestion of protein will still continue until we end up with amino acids. All right, so we have the brush border. So brush border is simply regions within the small intestine where we have a lot of villi and microvilli to increase the surface area for absorption and to complete the process of digestion. And we have aminopolypeptidases and dipeptidases. The function from their name indicate they break down larger polypeptide into the actual amino acid. So remember from the cell biology that we said the process of synthesis of amino acid involve the adding of amino acid in sequence and joining them to form polypeptide. Digestion reverses that process and make sure we end up with a 20 amino acid at the end of the day. So inside the enterocytes, as I said, we have the peptidases, and the function is to break the linkages and ensure we end up with amino acids. So for carbohydrate, what we absorb at the end is either glucose, fructose, or galactose. For proteins, we end up absorbing amino acids. So let's see what happened with fats, sometimes called lipids. So the digestion cell happen at the small intestines, and it start with the emulsification by the bile salts, and that one ensure we increase the surface area. And then we have the enzyme, which are the lipases, so specifically the pancreatic lipases. And we know that the fat is made up of triglycerides and fatty acids. So we make sure that at the end of the day, we reverse the process and break it down. So the bile cell, as I said, is involved in the process of emulsification. And so the micelle at the end of is formed by the bile salts, and it activates the process of digestion. So absorption, we need to increase the surface area. And I think I talked about this one initially, that we have a folding within the small intestine. And that folding increases the surface area by almost a factor of 10. And within it, we have the microvilli within the brush border. And this one gives us a very big surface area of about 250 meters square. So the small intestines, this is where water is absorbed, and the absorption is through the simple diffusion. And so the sodium is actively transported, and once we transport sodium, so the process of passive, that such that the water will follow it passively. But other electrolytes such as calcium, iron, sodium, magnesium requires an active transport. For carbohydrate, we said 80% will form glucose. So the absorption is through a process we call the secondary active transport. So I think remember from, your examination at the end of the first semester, where I gave you a question to distinguish between the primary, the secondary active transport and primary active transport. This is where it's utilized, where we have what we call the co-transporters. We have the sodium potassium co-transporter, and that is how sodium, and that's how rather glucose together with galactose is absorbed. For fructose, it's simply a process of facilitated diffusion with a little bit of endocytosis. And for protein, I said the amino acids is what is absorbed finally, and that one is also through the process of secondary act of transport. That is the sodium co-transporter. For fats, once we have digested them, so they have to be absorbed in form of monoglycerides. And so once we have the monoglycerides inside the enterocytes, so the fatty acids and the monoglyceride will recombine to form a triglyceride within the enterocyte cells. And the combination of the two of them will give us something called the chylomicrons. And the chylomicrons will be taken into the lymphatic system, and they will be transported until they are drained into the blood. And usually that one gives the lymph a little bit of the yellowish color because of the chylomicrons. We're almost the end. So after digestion, we have digested, we have absorbed what remain, and this will lead into the absorption of sodium and chloride, and absorption of water, so about 5 to 7 liters per day. And whatever remain now is a solid matter, which you call the fecal matter, and 30% of it is solid bacteria, and a little bit of fat that has not been absorbed as some organic matter, and undigested material, we call them the ravages. Usually bacteria will tend to break them down, but for human being, that aspect is not so much developed, unlike for other animals such as the ruminants that have quite a good type of bacteria that is able to digest and absorb the ravages. Well, in a summary, that is what the GITs involved. I know that this you could be feeling a little bit nauseated after we talk about the process maybe of defecation, but that is the end of it, and this is where we end. So the next section is about disorders, and pathophysiology. We'll do that one in the next semester.